Hello, I'm Ruben Sagar, and I'm very excited to present our work on evaluating the impact of forest disturbance in tropical rainforests through canopy science. The environmental conditions such as temperature, light, and humidity that organisms experience may be at a very fine scale, known as the microclimate, and can be quite different than the macroclimatic conditions. Such microclimatic conditions have been shown to affect species assemblages and functions in tropical forests. The forest structure, which largely determines these conditions, are affected by human disturbances, which impact the structure and will be reflected in the microclimates. The illustration on the right shows the air temperature and light intensity, two very important microclimatic variables, which are the highest at the upper levels and decreases as you go nearer to the forest floor. There is evidence that microclimates in the lower levels are buffered by the forest structure and hence much less extreme. But such knowledge in the context of forest canopies remains limited, especially in South Asia. Further, vascular epiphytes and beetles respond negatively to more severe microclimatic conditions through changes in physiology, life history, and distribution. Hence, in this study, we focused on vascular epiphytes, a typical but lesser studied component of rainforests, and beetles, one of the most abundant insect groups, to answer the following questions. First, we asked whether canopy microclimates differ between primary and secondary forest stands, and whether beetle and epiphyte communities differ between primary and secondary forests, and are they influenced by the microclimate? We focused our research in the southern Western Ghats, a biodiversity hotspot in India. We chose Kalakad Mundanturai Tiger Reserve, a protected area with primary forests and secondary forests, which were intensively logged in the 1960s and have been regenerating since then. We chose 18 trees to sample in both the forest types. We chose only Cullinia exarellata, a dominant canopy species common to both forest types. We randomly chose trees, but made sure they were safe to access using the single rope technique as shown in this image. In each tree, we placed one data logger for about 3 to 4 days, as well as 4 baited funnel traps, as shown by the image on the top right. We also estimated the number of epiphytes, as well as recorded species as much as possible. To answer our first question, we compared daily mean values of temperature and light intensity from the primary and secondary forests and tested them with the t-test. The temperature was significantly different, and the chart for hourly mean temperature for all the trees shows consistently higher temperatures in the secondary forest canopy. We also observed a similar trend for the light intensities, but it was not significantly different. We also looked at the frequencies in range classes of 1 degree Celsius for temperature and 1000 lux for light intensities. Lower temperatures and light were more frequent in the primary forests, and the difference was significant for both. The density plots show some portion of the ranges of the temperature and light. Next, we looked at the abundance, richness, and Shannon diversities of vascular epiphytes, which were higher in primary forests, but not significantly. The communities somewhat differed, as indicated by a Bray-Curtis dissimilarity index of 0.69. The microclimatic variables did not explain the variation in epiphytes as checked using a multiple regression. Contrasting to the epiphyte findings, beetles were consistently and significantly higher in the secondary forests, which we did not anticipate. The communities also differed between the forest types, indicated by the Bray-Curtis dissimilarity index of 0.76. However, a multiple regression analysis showed that only beetle abundance was somewhat explained by the microclimatic variables. This study shows that even after 60 years of regeneration, the microclimates in secondary forest canopies were nowhere near those of the primary forests. Beetles clearly preferred the secondary forest, although it is not clear why. But from a couple of previous studies from the region, we believe the beetles could be responding to presence of certain understory and mid-story species and not the absence of canopy trees. It is also quite possible that the surface temperature of substrates such as leaves and the branches are more important than the ambient air temperature. The response of the epiphytes was on the lines of what we expected, because larger and older trees with more complex branching has shown to harbor a more diverse epiphyte community, and maybe a larger sample size would have strengthened the results. We also show that light and temperature may not be the strongest drivers of beetle and epiphyte communities, and other factors such as relative humidity, canopy structure, and substrate surface temperatures and moisture may be more important. Nonetheless, being the first study of its kind in the Western Ghats, we believe this work begins to fill the knowledge gaps in ecology of forests in the context of microclimates, and this work has a lot of potential to be followed up by future research examining microclimates and biotic responses to environmental change. I would like to thank you all for your attention, and also the Rafford Foundation for funding this project, and ATRI and the Tamil Nadu Forest Department for facilitating this work. Thank you.